So let's get started with Arianna Bochinelli, who's going to talk about fish.
Okay, hello, I'm Arianna, and I'm going to talk about the first work I've done in the field of collective animal behavior, which was trying to answer this question: how fish use the movement of other fish to make a collective decision, which is a work trying to put together two approaches. One is about movement, and one is about taking collective decision. So why are we interested in fish collective decision? Because basically taking consensus decision is very important in human society, and how to find an agreement is a very important topic, and it's important in social science and have a lot of implication in economy and politics. But what people realize recently is that also animals living in groups are required to make a constantly collective decision, for example, about where to find food or to choose a new shelter. And also collective movement can be thought as a continuous decision-making process about speed and direction. And why it's interesting, because actually animal groups are often quite good at finding an agreement. And what's being shown is that the larger is the group, usually the best the group is to take a collective decision. So to take a particular example, there is a recent experiment about fish which shows explicitly that the accuracy to choosing a branch and avoiding a predator is an increasing function of group size. So in this experiment, they were putting this fish in this Y-shaped tank, and in one of the branches of the tank there is a replica of a predator. Then they were observing fish moving toward the branching point and choosing branch, of course. What they observed is that one fish alone is very poor to finding the right branch, but with the increasing number of the fish in the group, they were able to take better decisions. So here, what you see is that nobody's coming back. What they were able to see is that this accuracy, that is the probability of spotting and avoiding the predator, is an increasing function of group size. So here, blue spots are data. And this trend has a nice theoretical explanation in the many-eye model, stating that it's enough that one fish spots the predator in order to the whole group to avoid it. And this is due to the transfer of information within the group. And this can be translated to a formula, and it is a nice fit. So this sounds like a nice explanation. And what one would expect is that the larger is the group, the better is the decision, but also the longer is the time to converge to such a decision. But what they were able to see again in this experiment is that also the speed of fish increases as a function of group size, while the virtuosity, which is a measure of how not straight the fish are going, is a decreasing function of group size. So basically, larger groups go faster and straighter, and they can take better decisions, which is pretty remarkable and can be explained with the fact that the information transfer is mediated by movement in fish, and this thing allowed the information to spread very quickly to the group. So that's why we were asking which are such movement interactions that can allow for very good decision and very quick decision. We tried to find an answer by using a very classical approach, which was choosing a model and simulate this decision process in order to derive the minimal set of rules allowing for fast and accurate collective decision. So now I'm going to talk about the model. The model is a self-propelled particle model where fish are described as particles and characterized by a constant speed in some direction and by an interaction zone, which is this gray area. So we are talking about about a metric interaction. This interaction zone defines the interacting neighbors, who are the interacting neighbors. So my fish is going to interact only with the fish inside the zone and ignore who's outside. And then the movement of the fish is a consequence of different contributions that are represented as vectors, and the length of the vector is proportional to the importance, to the weight of this contribution to the movement. So what we have in our model is an inertia in the current direction of movement. Then we define an aiming force, which in this case we point toward the branching point of the tank. Then we added an angular noise, and the social interaction, the first social interaction we talked about is attraction toward the mass center of interacting neighbors. But then we had to also add an alignment with the neighbors, because during the work we realized that it was not possible to fit the data only with this attraction toward the mass center. So at each time step, all these contributions are summed up, and they give the direction of movement of the next of the following time step. So once defined the rules of motion, we made a numerical simulation of this awareness experiment. Here there is a very rough visualization, but anyway, the fish starts swimming from the starting point of the bottom of the tank and go toward the branching point with under this aiming force. And at a certain distance, which is taken from data, they have a spotting probability, and each fish has this spotting probability, and the fish spotting the predator will stop behaving according to the rules I explained, and will go straight to the safe branch of the tank. So what we measure in this simulation, and we want to sit against data, is accuracy, so the probability of the whole group to avoid the predator. The tortuosity, which is a measure of how the path of the fish is curved compared to the straightest path between 2 points. And the standard deviation, which is a measure of how many turns, more or less, the fish does during the decision process. I will skip basically the details about fitting, but the main point here is that we fitted for just one fish. We fitted the tortuosity and circular standard deviation. Indeed, the accuracy is trivial, and we got these parameters value for the aiming force F and circular error E. And then we plug these results in simulations of an increasing number of fish, which is the same number that they were using for experiments. And we fitted the accuracy and tortuosity, so color dots are experiment, and the boxes are the best fit we could find. And then we got the values also for the attraction toward mass center and the alignment. Let's forget about the exact value, but the point is that the main finding here, that what we got is that alignment appears to be more important than attraction towards the neighbor in order to have this decision-making process. And another thing is nice to notice is that we were fitting against accuracy and tortuosity, but with those parameters value, we can get also the correct trend for circular standard deviation. So once we have our values, we can relax a little bit and see how the experiment was looking like. So here is a group of 8 fish. I don't know if you see them. They should come. Come on. Nothing working here. There should be a group of 8 fish coming up and spotting the predator.
Can I just ask you, what is the predator?
No, that's just from the...
Yeah, but I mean, what do you put in that
Oh, that in the experiment it was the replica of a predator.
So it's all model?
Yeah, because there are problems with ethics. They cannot use an actual predator. Yeah. No, I don't know. Moving on. That's bad. So here is. So here we go. Okay, here we have the fish. So they go toward the branching point, they look around, and then one spots the predator, and everyone chooses the right branch. So that's how the experiment looks like, and we can compare to how, I think it's this one, I'm not sure. We can compare with how the simulation looks like. So here we have fish. I hope they're going to, yes. Okay, the one becoming redder is the one spotting the predator. And then the other one keep on behaving as with the same rules, and then they follow the one spotting the predator and choose any of the same the correct branch. So the last thing, the last.
Could the first fish not spot the predator?
Yeah. Yeah, that's the point. I don't know.
In the model, how is that possible?
Because once they get here, like this line is more or less the area where they were seeing an experiment, starting to understand they had to choose between two branches. So here, each fish has a single probability of spotting the predator, but it's not necessary that it will spot. So in this case, for example, like the one getting red is the one spotting. It's not the first one. At a certain point, there is one spotting the predator, and then it gives the right direction to the right branch. And everyone else follows this rule. And
what if the first fish is wrong? What if the first fish missed the right one?
And it gets eaten.
Okay, like that. What about
okay. Yeah, they were observing some sequentiality. So if the first ones don't spot and choose the wrong branch, it's more likely that they will follow. It's a kind of leading behavior. But this kind of fish, this species of fish, they are schooling fish. So they are not likely to follow a leader. They are more likely to swim together according to some rules. So they will take into account what's going on around. I have still a little bit to show, but I should. So last part of the work was about asking, okay, now we have these rules of motion. We found which are the interactions that are likely to explain how this works. That are likely to explain this experimental outcome. But this is a very particular situation. So we asked, okay, this is kind of a video game, we have more serious stuff. But we asked, okay, what about the different environments? So we modified the slide of the model in order for fish to behave according to the same rules of motion, but in different conditions. That's what happens if you don't as well. And then try to predict, try to see if predator avoidance, the same rules of motion can predict successful predator avoidance increasing with the group size. And except for video games, the result seemed to say yes, because we were measuring the percent of escaping fish, the total percentage and the percentage escaping just due to interaction, so the fish should even spot the predator. And we found that this is an increasing function of group size as well. So this seemed to suggest that this same interaction might be relevant also in open water and in different environments. So to sum up and conclude, we were asking about the minimal set of rules of motion that could reproduce the experimental outcome, that is fast and accurate collective decision. And we tried to answer through a modeling approach, and what we found was that we need both alignment and attraction in order to fit data and reproduce the experiment. And also that alignment with neighbors is more important, turns out to be more important than attraction. So the following analysis was in a different environment, and this analysis seemed to suggest that also in open water the relevant interaction could be the same as in this particular environment of the experiment. And future work about this would be to try to plug some new rules of motion that were fitted and inferred in another recent paper for the same species of fish. So this rules of motion predicts actually that attraction is more important than alignment. So it would be very interesting to plug them inside this white maze experiment and see if they can reproduce the decision-making process as seen in the experiment. So if I still have a minute, I will present the people I work with for this project. In Uppsala, there is David Sumter, who's my supervisor, and then Cavagna and Dennis Stromholm. We work basically on model. While there is much more lucky people in Sydney doing experiments on fish, Ashley Ward, James Ibertrid, and Jess Kerr in Berlin. So thank you very much for your attention.
Time for one question.
Is there an element of learning each time? Do you do the experiment multiple times in the same space and do they learn at all?
No. It was not me doing the experiment, but I'm pretty sure they were avoiding this learning thing. Yes.
What steps did you take to ensure that the fish are not responding to some other cue? Like, for example, you said you used a fake predator and other design things.
Yeah, so the idea that they go straight to the branching point is because they are responding to the fact that the two branches of the white maze, there is sand while the tank is white. So they are swimming there because they are scared by the fact that they are on a white tank and they want to go to the dark soon. So that's why they swim there. And then they choose a branch because of the replica of the predator, which is a replica pretty realistic. That's what they say.
Is it well established in nature that the fish like realize that there's either biovision or like do fish in the wild like see predators by vision alone, or is it that they detect some other?
Oh yes, they also have lateral lines to interact, not only visually, but those kind of fish, they rely more on vision than on lateral lines. So that's a good experimental part, and I'm not very. And I just know roughly that they were doing experiments like trying to not make fish not using their lateral line, and they were seeing that they are good at vision, those kind of fish. Then what else? Another thing that might be interesting is that there are also experiments with a white maze tank but without the predator, and they show that there is not preference like the right or left, then it's the same thing.
Thank you.
So we move next to Anna Mellinger, who's going to talk about cooperation and how it evolves and emerges in the populations.
Yes, thank you. So I want to talk about this cooperative material, and the work was done in Munich under the supervision of Erwin Klein, and Jonas is also here, so if you have any question you can ask about that. So we wanted to investigate the reproduction of cooperation bacterial population, and so the question is first how do bacteria cooperate? So they mainly cooperate by the production of a public good. So a nice example here is Pseudomonas aeruginosa or Pseudomonas bacteria, and what they need for their metabolism is iron. And iron is usually like present in large clusters, so it's hard to get it. And what they do, so what some of them do, the cooperating bacteria, they produce a public good, as cited before. This is a small molecule with a really huge binding affinity for iron. So they release this Tidal 4 into the environment. There it dissolves single iron molecules, builds up a Tidal 4 iron cluster, and this diffuses around, and then the bacteria can eat it and use iron. The problem which now arises is that only the cooperating bacteria pays a metabolic cost, so they need some of their energy, and they reproduce slower than the free-riding materials. But all of the bacteria can use the iron. So we have kind of a dilemma of cooperation, which means that like, it would be optimal if everyone would produce it, but the cooperators can be always exploited by the free riders, and in the simplest terms of Darwinian evolution, cooperators would die out. Well, this is actually a true nature, that's why I wanted to try to analyze this. So there are some ecological factors which help cooperators, and the first one is population growth, which is pretty obvious. So if you have a population with more cooperators, there's more public good present, so the whole population can grow better. And this is shown here. These are some experiments for the motility. What you see here is the total population size and how it evolves over time, and the different colors correspond to different initial fraction of cooperators. So here at the top we have colonies which grow many cooperators, which grow fast to a large size, and this declining fraction of cooperators grows as hampered. So in principle this is an advantage which could overcome the selection disadvantage. What we need here to make this advantage work is population structure, because only if you have several colonies where some have more and some have less cooperators in it, you can get like, you can get a positive effect to these cooperators. So we need population structure, and we need something else, because when you think of a single colony, in each colony the cooperator grows slower than a free rider. So in the long run, cooperators will die out, and if we don't have something additional which restructures, which forms new colonies, where this growth range can play again, cooperators will also ultimately die out. So, so this is in principle a nice theoretical thing that we think, okay, if you have a restructuring mechanism, we can make it somehow work. The good thing is that this restructuring is really common in nature. So this is just a typical bacterial life cycle where we have bacteria which form some colonies. So they start from a small initial population size. Then this colony grow, and in the growth phase the public good is present, and also cooperators. They are also reproduced, and after a certain time the colonies disperse and it restarts. So in principle, this happens really for many bacterial strains in nature, but in principle those life cycles are highly regulated, really complex. And we here don't want to go deeper into all this regulation mechanism, but we just wanted to investigate the principal influence of such a restructuring mechanism on the evolution of cooperation.
So what's the difference between the white and the dark bacteria?
I think so. In this example, the outer ones kind of shields the inner ones when the nutrients get rare. So this is so often this process happens after nutrients get rare, and then the first try to save them, and after a certain time they blow up and build new colonies. So what we really wanted to do was just wanted to have a simplistic model of such a restructuring mechanism. So what we have an initially well-mixed population consisting of cooperators and free riders. Then we form some small colonies at random, so they're formed according to the initial fraction of cooperative X naught and N naught. And then we let these colonies evolve for a certain time, and after a certain time the cycle restarts. So in principle, during this evolution, there are just two important things for you to remember. So the first thing is that in each colony, a cooperator has a growth disadvantage because it reproduces a public good, which is metabolically costly. The first thing. The second thing is that in colonies containing more cooperators, more public good is present, and therefore also the colonies grow better. So so in the next slide, I'm going to tell you how we really modeled this evolution of the colonies. And this we did so standard, many of the standards approaches for this population evolutionary dynamics have a constant population size. This obviously doesn't work for our model because the advantage comes in with growth. So what we needed is we modeled this with independent birth and death rates. And in these birth and death rates we included the features I talked about. So we have first a class with G, which doesn't depend on the species as it doesn't depend on whether it's a cooperator or a free rider. And this G has to account for the advantage due to the presence of public good. So the more corporates are present, the more public good is present, and therefore the better the bacterial growth. So you see somehow monotonically increasing function of the fraction of corporators, and what we choose here for specificity was a linearly increasing function, but our results don't depend on the exact form. The second thing is that we have corporators and free riders, and as I told you, corporators rapidly slower than free riders. So this is in this species-specific parts here. So corporators have always growth rate which is slightly smaller than the one free riders. And then the bacteria also die. So, and we assume here logistic growth, which means there are not infinitely many nutrients present. So if the populations get huge, the death rate increases because there are some metabolic products which poison each other, and there are also the nutrients which kind of hampering factor. So to get an idea, so we can build a master equation with this, and to get an idea what the dynamics really do, let us have a short look at the deterministic equations. So the first equation shows how the fraction of corporators deterministically, so in a method approach, evolves with time. What you see here, so we have nothing gained, so it's still negative on the right-hand side, so the fraction of corporators without any fluctuation, that's important, dies out. And this equation is also looks pretty similar to the standard equation in which this corporation dilemma is described. And the second equation here describes the revolution of the population size. And this equation resembles the equation of logistic growth. You have like an exponential increase at the beginning, and then it separates to a fixed value. But here something is a bit different. So we have a carrying capacity, so a maximum population size, which now depends on the fraction of corporators, which also makes sense when you have when you think about the second slide where I show you the growth curve. So actually the more corporate populations can go to our other side. So let's now have a look what's happening in the group evolution step. So we have several colonies which evolve, and what I show you here is the time evolution. The black lines are the solutions of the deterministic equations I just showed you. So here's this for the population size, we have this exponential growth at the beginning, and then it separates to a fixed value, which then declines because the fraction of corporator declines. And so on the right-hand side, you see how the corporator fraction evolves over time, and deterministically it only declines. But this changes if we have a look at the full stochastic solution of this problem, because then this appears. So what you see here is that the behavior changes. So the fraction of corporators first has a kind of transient increase, and then it saturates to a fixed value. And so in the following, I want to explain why you have this transient increase and why it separates to a fixed value and corporators are not out. And one thing I want to mention here is also that you see that the advantage of corporators gets huger with small and not. So it's a fluctuation effect. The small and not fluctuations get larger, and therefore also the advantage of corporators get larger. So let me first turn to this transient increase in the beginning. And intuitively you can understand this easily. So at the beginning the colonies grow like exponentially. And when you now consider several colonies in a population of some with more corporators, some with less corporators, and the colonies with more corporators just grow faster. So what you have is when you average over the whole population, the colonies with more corporators have a larger weight. So you have kind of a positive covariance between X and N, and this is the reason why we can have this transient increase. And you can also see this in this movie. So what you see here is the probability distribution for the population size and the fraction of corporators. And in the beginning you see we've kind of a broad distribution which colonies can contain different amounts of fraction of corporators at the beginning. And when I let it run further, you see that the colonies with more corporators grow to a much larger size, and this makes this transient increase. This increase is just transient because in the long run, corporators have the selection disadvantage that they are out in each colony, and therefore it goes down again. So one important thing is the parameter dependence of this effect. So first thing I told you, it's a fluctuation effect, so it's getting stronger and smaller enough. Second thing, which is not so intuitive, but you can see it when you calculate the whole dynamics, including the fluctuation, then you see that the effect gets also stronger with a small x naught. So for really just a few cooperators at the beginning, you get like a huge maximum, while the maximum gets smaller and smaller for a larger initial fraction of cooperators. And this will turn out to be important. So it's the second mechanism, which emerges in the long run, is even easier. So when you again think about the formation of different colonies, we have like colonies only containing cooperators, only containing free riders, and everything in between. And in the long run, as I told you, cooperators die out in all mixed colonies. So we end up with such a picture. So we have most of the colonies just contain free riders because they had the evolutionary advantage. But there were some colonies which contain only cooperators at the beginning, and these colonies can evolve without being exploited by free riders. So these colonies get really huge, and again we have a shift in the weights, and this can also kind of cause stable level of cooperation in the long run. The difference is that here, so the strength of this effect only relies on the probability for having a purely cooperative group in the beginning. And this probability gets higher with large x naught. So the dependence on the x naught is just the other way around, as in the first effect. So before coming to seeing what's happening under the regrouping, I just want to show you briefly that we have some experimental data which Heinrich Jung measured also in Munich. So what he did is he used Torremolinos Puritas strain, and he in principle subdivided it in several colonies, and then he measured over time. And what you can see is in principle that you also have this transient increase at the beginning, and then it separates to a fixed value. So we are kind of happy about these results because they really resemble our stochastic simulations. So with this at hand, let us now look what's now happening if we regroup the thing. So we were here, we had in this time, so we have this time dependence, and now as there are two effects, we have the transient increase and the stable level of cooperation at the end, it should matter if we regroup here or here, because we have this time dependent dynamic. And let us first have a look what's happening if we regroup for large regrouping time when we are already in the stable regime. And this is shown here, so this is the fraction of cooperators, how it revolves over several of those regrouping steps. And what you see is kind of a threshold scenario, or you have an unstable fixed point here. So if we have a cooperator fraction which is below a certain threshold, cooperators die out. But if they are above a certain threshold, they take over the whole population. And the reason for this is just that they're the X-naught dependence of this fixation effect. So only for large enough X-naught, it's strong enough to overcome the selection disadvantage, and then cooperators take over. But below this, they die out. This changes if we regroup for small regrouping, for small times, so in this transient regime, because then the effect is stronger for small X-naught. So when you get to the stable fixed point, you see if we start with really tiny initial fraction of cooperators, the slope is the most pronounced, so we have a strong increase, and so it doesn't matter at which initial fraction of cooperator we start, we always end up at a kind of coexistent fixed point where cooperators and free riders live together. So this is pretty nice because it also would allow for the evolution of cooperation from a single mutant. So you can think about really that there's a purely free riding population, we have a mutation that a cooperator emerges, and this cooperator also has a reasonable probability not to die out immediately, but to kind of survive for a long time. So at my last slide, I just want to give you the full picture, but in principle, so here's here shown the two fixed points I just showed you. So we have the stable fixed point. So for against the regrouping time, so if you regroup for small times, we got this stable fixed point with coexistence between cooperators and free riders. But for larger regrouping times, we get out of the regime where we had this transient increase as maximum, so this fixed point vanishes, and instead an unstable fixed point arises, which belongs to the threshold scenario, and only above this threshold, cooperators can be present in the population. So, okay, sorry. This I already told. So with this I want to conclude. So I hope I have you convinced that kind of population dynamics is also important for evolution, because many of the evolutionary models neglect it and just assume constant population size. And this effects here really relied on the fact that the populations grow differently with different fractional cooperators. And what we could show was that due to this growth differences, and due to the fact that there are strong stochastic fluctuations due to small initial population size, cooperators can have a reasonable advantage in the population, and the advantage can be even strong enough to have a single cooperative mutant or just a few like spread into population and to maintain cooperation. So last, I want to thank you for your attention, but I also want to thank Jonas, who did a huge part of his work here, and Erwin Frey, who supervised all of us, and Heinrich Jung, who did this nice experience. Thank you.
In nature, is there always a clear distinction between a cooperator and a deflector? So in nature, is there always a clear distinction between these, or is it going to be something that can flip?
So, yeah, there also exists these scenarios where you have like a phenotypic heterogeneity, where the cooperators do not always cooperate, but it looks, what we wanted to do here, we wanted to have a look at the worst case scenario, because if a cooperator has to cooperate, then it doesn't matter whether it's good or not for him. So then it's really the worst case, and when you explain that, can explain there why cooperation is there, it's even better in the heterogeneous part. But we're definitely
Would this be an interesting system for doing inkjet printing of cooperator colonies versus non, so you get very specific geometries and study boundary effects, or is that not really?
That's not our. So what we want to do first. So the nice thing really about the system is that the public good societal for is green. So we can. The nice thing is you can really measure what's really happening with societal pro, and that's definitely the first step now with this experiment. And then you can further. But so the first thing is really to understand how really this public good is regulated and how it's really going on.
Are there scenarios where the cooperation bacteria actively fight against free riders, like the toxins or something like in the immune system?
Well, so this fighting thing, I'm not sure. I know that they can avoid them. Yeah. So if you have a kind of more cooperative clusters, so then they can't be exploited so well. So you can say it's like hidden discrimination, so they try to avoid the contact. Yeah, but for sure there are many mechanisms coming onto this. So this is really like the worst case, and all the assumptions you can make with that. Okay, worst case for cooperators, and we can. But there are plenty of this biology, you know.
Any questions? So about what are these mechanisms for grouping? So you didn't really mention that. How do they actually group?
So what we just did is that we mixed the whole colony and then we formed new colonies. In principle, you can think about so what I really have in mind is that you have like new colony formation, right. And then the bacteria from different colonies which group on, then they occupy new spots. But when you have something like, so when you do it purely at random, you have again like a worst case scenario, because when they come, for example, more from a more cooperative colony, they are again closer together, and everything which makes like the variance huger is better for the cooperators. So, yeah.
So what happens when you fluctuate the grouping? Does it matter if you fluctuate?
So we also try it, sure, yeah. So in principle, you can then jump between those two regimes, and then you can think about some pathways that you have. So it's beginning just a few cooperators, then you have a sequence of short group times they spread, and then they are above the threshold, and when then longer, they can overtake. So in principle, all of those stuff then can happen. Yeah. So we did some simulations of this, but in principle, this is also kind of more beneficial for cooperators too.
Thanks. So we move on to the next speaker, who's an ISC-CNR. She'll be talking about the cloning spreading of bacterial colonies in biofilms.
Thank you very much. I'll be talking about biofilms and some work that I've been doing with several people in Harvard and Cluster. So this is a biofilm. This is the guys that we are working on. It's a Bacillus subtilis, which is a gram-positive bacterium. It lives in the soil. There are about 10 to the 9 cells in this colony. It's about 5 millimeters in diameter, and it's less than a millimeter thick. We grow them on agar, so what you see here is agar. The real-life biofilms are much more disgusting than this. They grow pretty much everywhere you don't clean, like in the bathroom or in the kitchen. They grow in the teeth, they infect everything, pretty much every surface in your body. So they cause a lot of problems. And that's why many people are interested in biofilms. Essentially, in this protected one of the main observations is that in this protected colonies, they become more resistant to antibiotics, and that's why they cause the infections. From a more fundamental point of view, they are very interesting because each cell here, well, not each cell, but cells in this colony, although they might come from the same cell, they are different. So this colony is differentiated. So this strongly resembles things that happen in developmental biology, and with a lot of caveats. The first one is that this is a transient multicellular organism, because it gets together, it stays together until the conditions are right, and then it gets dispersed. So cells get back in catatonic state. And in fact, if you take any of the cells and you put it back in liquid, it will grow happily for a long time. And the second caveat is that we don't really know what the wing is for biofilm. There is no real function that we know about. So instead of talking about, before talking about a developmental project that gets you to a biofilm, I want to tell you something that the biofilm does, which is expanding on surfaces. So here is a cartoon of how a biofilm happens. This is a surface that we have prepared for the biofilm. We have dropped one or a few cells on top of the surface, and if we are nice enough and we provide enough food and water and there is enough oxygen in the atmosphere, then that cells that we have dropped there is very happy. And what it does is it divides. And what it produces is identical donors. But then the interesting part happens, something in the environment becomes stressful, and the colony decides to become a biofilm. In this transition, what happens is most cells lose their flagella, and in fact, almost all of the cells in the first 24 hours start to produce a matrix. So I'm going to talk a lot about the matrix, which are these red cells. What matrix is a bunch of polymers, and it's a pretty general feature of many biofilms. In Bacillus subtilis, we know a lot about these polymers. So, in fact, you might imagine that because most of the cells lose their flagella, this biofilm does not move at all. And probably the second line of any paper that you read on biofilms will say that biofilms are sedentary communities that live attached to surfaces and in fact, if you watch these communities, well, for sure the velocities are a lot reduced, but if you watch these communities for long enough, you'll see that they expand on surfaces. And not only they expand, but if you look at close enough and you make a movie of what happens here, you'll see that cells actually slide on top of the other. So they are actually pushed in some way. Not only this, but things change depending on the strain that we use. So we know a lot about the genome of these guys, so we can knock out many things. If we knock out flagella from scratch, well, we know that not many cells have flagella in the biofilm. If we knock it out from scratch, nothing happens, essentially. The colonies grow in time. Sorry, I didn't write the times. This is 6 hours, 12 hours, 24, and 38. So nothing much happens. Flagella don't seem to play a big role in this expansion. But then, if we knock out the matrix, as you might imagine, a lot more interesting things happen. Here you see the plot of how the radius of these colonies grow in time. this is simple enough that even physicists can do. This is the wild type, the blue line is the flagella knockout, and this is the matrix knockout. So you see that there is actually a very nice separation about that. So the question that I'll try to answer in this talk is, why does matrix help in spreading? And just to give you an idea, this is, at the beginning it was very puzzling because this matrix, as I said, is a bunch of sticky polymers, so it was not immediately obvious why that would help in spreading. Okay, so here's a mechanism that we are proposing that is based on polymer production. So imagine that you are growing a biofilm. Imagine to sort of freeze your cells for a little bit so that they don't produce any biomass. Here's the agar. This is the petri dish, and we seal the petri dish. If we wait long enough, it will go to equilibrium. So equilibrium from the point of view of the polymers will be constant concentration, such that the osmotic pressure in the agar and in the biofilm will be the same. But then what cells do in the biofilm is they produce biomass. So they will drive the system out of equilibrium by introducing a fluctuation in polymer concentration. And so what the system wants to do, so here phi is the polymer concentration, here is the fluctuation, and this is the osmotic pressure that depends on the phi in some way, complicated. Okay, so what the system wants to do is it wants to go back from equilibrium, and the way it does it is water flows from the agar to the biofilm swells, and it changes shape. Now in this process, two things happen. One is the forces can be produced, and the second one, the biofilm changes shape. Okay, so we need a theory to understand how the shape changes, because of course you can imagine extreme scenarios where the biofilm just grows vertically, and remember that the food comes from here, so that wouldn't be a very smart thing to do. And on the other hand, if it expands horizontally, its nutrient uptake will increase a lot. So we're asking what's the evolution of the shape of the biofilm as a consequence of this swelling process. Okay, so I'll tell you the starting point and the end point. The starting point is we assume that the biofilm is essentially a mixture of water and biomass, and we'll assume that we are not far from equilibrium. So we'll write this function, which is the dissipation and the free energy change, and that will depend on the friction processes coming from water, biomass relative motion, and the stresses generated within the biomass and within the water. Then there will be a free energy change, depending on a change on the concentration, and this will enforce incompressibility of the total velocity. Okay, so from here, what you have to do is you minimize this functional with respect to the velocity of the biomass and of the water, and then you do some more manipulation. You assume that the biofilm is much larger than thick, and you can build a self-consistent thin film approximation for H. H is the shape of the biofilm. It depends on R. R is the coordinate, so it's 0 at the center of the biofilm, and it's big R at the edge of the biofilm. This is written in non-dimensional form, and what you see, a couple of things about this equation are this. First of all, there is just one non-dimensional parameter, K, so it's a very simplified equation where we can understand qualitative behaviors, and there are two terms. This term is given just by growth, and this term is an horizontal flux. So if K is very small, at least at the beginning, what we have is exponential growth in the vertical direction, given by the equation where we neglect this term. But in fact, because this term grows much faster than the others in H, eventually this term will become the same order of magnitude as the other. So it will take over, and the biofilm will start spreading. Now this is a little bit hand-waving, but you can make it much more precise. There is a self-similar solution to this equation. You can do simulations, and then what you get, this is an example of a simulation. This is the profile in time. This is the center of the biofilm. This is the edge. And the way I'm tracking the shape of the biofilm is by looking at this point. This point is the point of maximum slope in the biofilm. track the position of this point, which we'll call R, the radius of the biofilm. It's in fact the radius of the bulk. And we'll track also the shape, the slope at this point. So what you see, long story short, is exactly what I told before. You have an initial stage where you have no spreading. So that point stays fixed in radius. The radius doesn't change. And the slope increasing, because this is the exponential growth in the vertical direction. And then eventually what you have is that the flux term takes over and the biofilm starts spreading and at the same time smoothing out. So the maximum slope decreases. Okay, so we have a mechanism and a theory and we have this prediction, and now what we do is we go back to the experiments and see how it works. So what we did is, these are data on a real biofilm. The way we got it is we measured the transmitted light and then we mapped it to height by calibrating with side pictures. And these are the profiles at different time points between few hours and 18 hours. And here I'm marking exactly that point that I was talking about, the point of maximum slope. And what you see qualitatively is that at the beginning, this point is rising up and the slope is increasing. The biofilm is growing vertically. And eventually, that point is spreading out and smoothing. So we have the qualitative behavior that we expected. Now, if we redo the same thing with the APS knockout, which has a dramatically decreased concentration of polymers, what we have is this picture. So this is, again, the center of the biofilm, the edge of the biofilm, same time points. What you have is the point of maximum slope doesn't move at all. So it stays here. The biofilm keeps sticking. We can make this quantitative because we have numbers and we have two parameters essentially. One parameter is the division rate, and we get it from, we infer it from volume growth. We'll assume that the division time is constant. You see that it doesn't change much between the wild type and the matrix mutant. The average division time is 2.3 hours. Then we have a prediction for the slope, and from this prediction, we'll infer this one fitting parameter k. It's in fact very small, so it makes sense that it starts with the exponential growth, and it takes a while before the flux takes over. So we have this number for this. Sorry, this is the wild type, and this is the APS knockout. So we have this fitting parameter k, and now we end up with a prediction that has no free parameters, and we superimpose it with the data for the radius of the biofilm. And we see that the wild type agrees pretty well, while the EPS knockout doesn't agree at all. So this is essentially what I wanted to tell you today, and I'll say a couple of comments. The first one is that we were happy about this result because it starts to give an explanation why the EPS knockout is less fit. So if you take the EPS knockout and the wild type in shaking cultures, they are perfectly fine. They have the same division time so there is not very much of a difference between the strains. But when you put them in a biofilm and then you count later how many cells they produce, you'll see that the wild type produces much many spores, and all of these are matrix mutants with different defects, and they're all less fit. And also the number of cells produced by the EPS knockout is much less. And people were very puzzled about this because producing EPS is costly metabolically, so there is no a priori reason why you should be less fit if you don't produce it. In fact, it should be the inverse. Okay, so this was the main result that I wanted to tell you about. What we're doing now is trying to see and if there is any biology behind this. Because of course, in the model, I didn't give you many details, but I assumed that everybody was producing the same amount of polymers, and all of these boring situations where there is nothing happens in the environment. But in fact, the cells are different and they can make different decisions. So are they aware of the power that they have? Are they using this mechanism to go somewhere? So what we are trying to do is trying to see whether the biofilm is able to translocate somewhere on a surface, if there is any reason to do that. So if you put gradients of different things. And last, the other directions that we are going is this one. So I was mentioning before that one of the reasons why people are excited about biofilms is that they strongly resemble multicellular organisms. And this is one of the reasons why. So these are the profiles. This is a section of a biofilm that has been frozen. And different genes have been tagged with different fluorescent proteins. And you can see that there is a development of gene expression. And this picture is pretty much conserved. You can do it many times and the same numbers will pop out. This is just a longer section. So what we're trying to do now is trying to see how the biofilm is able to control the spatial temporal pattern of gene expression. And with this, I conclude, and I thank you and everybody.
So in the previous slide, we were showing the new experiment. What breaks the symmetry? Why is there expression on one side and not on the other of certain genes?
This is one of the points. It's actually not my experiment. These guys did it before we started to work on this project.
But there were boundary conditions. Are you showing the sections of the boundary?
No. This is a vertical section. You have the biofilm.
Yeah.
You cut it. This is the center of the. Actually, it's not even the center. The center is here. So this is the very edge.
Okay.
So this is the height. Here there is the agar. There. Center edge of the biofilm.
Yeah.
Yes.
I understood that, but I just don't understand why what induces this asymmetry is the fact that they started growing in the middle, so they know what is outside and what is inside. And also when you were showing me pictures of the agar, the middle spot was always darker.
So there are two different questions. The central spot, that's the spot where you put your cells initially. And there are studies showing that there, because it's older, iron is missing and a couple of other things. So it looks different because of that. Because of course, in the middle you have less stuff than at the edge. Stuff comes from here, has to diffuse, so there is a gradient, right? And the same sort of goes for here, but we don't really know. So we know a lot about the signaling cascade that gives you the matrix production, but because there are a lot of interactions and a lot of molecules, signaling molecules that are produced, emitted, and trigger other cascade, we don't exactly know why this happens. And this is sort of like why we are into this business.
If you have an asymmetric starting spot, let's say a half circle rather than a full circle, do you get an asymmetric biofilm?
So I think eventually if it's not too big of an asymmetry, you recover the spherical symmetry. There are people doing like lines, and that of course that changes completely because it's just diffusion and reaction of the nutrient. The nutrient comes from below, then you'll have a different pattern to start from a different condition.
Other questions? No? Okay. Thank you. And we have like